In this nervous system part 3 video, we will cover protection of CNS, blood-brain barrier, ventricles. Protection of the CNS. The brain and the spinal cord are delicate structures surrounded and protected by an outer covering of skin and bone. Cranium is protecting the brain and vertebrae is protecting the spinal cord, and by layers of connective tissue called meninges and by cerebrospinal fluid. The meninges are 3 layers of different types of connective tissue that cover and protect the brain and spinal cord. The first layer is dura mater. Dura mater is the outermost layer, is made up of fibrous, tough, white connective tissue. It is a strong outer sac that encloses the brain, spinal cord and the cerebrospinal fluid. It has many blood vessels and nerves and attaches to the inside of the cranial cavity. It also extends inward between the brain lobes to form protective partitions. It continues into the vertebral canal to surround the spinal cord, ending in a sac at its end. The dura mater has 2 layers of fibrous connective tissue. Its periosteal layer, which is more superficial, attaches to the periosteum, the inner surface of the skull. Around the spinal cord, there is no dural periosteal layer. The meningeal layer actually covers the brain, continuing caudally as the spinal dura mater in the vertebral canal. The 2 dural layers of the brain are fused in most areas. In certain places they separate, enclosing dural venous sinuses, which collect blood from the brain and channel it to the internal jugular veins in the neck. Next is arachnoid mater. Arachnoid mater is a delicate, cobweb-like layer lying beneath the dura mater that contains most of the blood vessels. It is the mid layer. The subarachnoid space separates the arachnoid and the pia mater. It contains delicate connective tissue through which cerebrospinal fluid circulates around the brain and the spinal cord. The innermost layer is called pia mater that follows the contours of the brain and spinal cord and adheres tightly to the highly folded surface. The meninges continue down within the spinal cavity for some distance below the spinal cord. The thin pia mater has many blood vessels and nerves that nourish the brain and spinal cord. The pia mater is closely aligned with the surface of these organs. It is comprised of many tiny blood vessels and delicate connective tissue. There are also spinal meninges, discussed later when we cover the spinal cord. Meningitis is the inflammation of the meninges. Next we will see the cerebrospinal fluid. CSF is produced by specialized ependymal cells in the small red choroid plexus of the ventricles of the brain. A bed of capillaries, that is small blood vessels called a choroid plexus, exists in the walls of the ventricles. Most CSF is formed in the lateral ventricles. Ependymal cells cover the capillaries and take what they need to make CSF from capillary blood. Arachnoid villi are knob-like projections that protrude superiorly through the dura mater into the superior sagittal sinus. They absorb CSF into the venous blood of the sinus. In adults, clusters of arachnoid villi form large arachnoid granulations, where CSF is actually absorbed into the venous circulation. CSF also enters the meninges subarachnoid space via the 2 lateral apertures and the single median aperture and is reabsorbed into the blood. CSF surrounds the brain and spinal cord, maintaining a stable ionic concentration and protecting CNS structures. CSF is a clear, colorless fluid that circulates between the ventricles and the subarachnoid space to bathe the brain and the spinal cord. The cilia of the ependymal cells, gravity and pulsation of arteries in the brain are responsible for the circulation of CSF. Approximately 500 ml of CSF are produced daily. The same amount of CSF reabsorbed through arachnoid villi back into the bloodstream every day. So there are only approximately 100 to 160 ml of CSF present at any one time. Cerebrospinal fluid is similar to blood plasma but contains less protein. CSF contains more sodium, chloride and hydrogen ions but less calcium and potassium than blood plasma. CSF circulates within the ventricular system of the brain. The majority of CSF is produced within the two lateral ventricles. From here, CSF passes through the interventricular foramina to the third ventricle, then to the cerebral aqueduct, then to the fourth ventricle. From the fourth ventricle, the fluid passes into the subarachnoid space through four openings. The four openings are the central canal of the spinal cord, the median aperture and the two lateral aperture. CSF is present within the subarachnoid space which covers the brain and the spinal cord and stretches below the end of the spinal cord to the sacrum. Now let us see the important functions of the CSF. The CSF allows the brain to float in the cranial cavity. Without the CSF, nerve tissue would be damaged by the sheer weight of the brain against the bony floor of the cranial cavity. Then the CSF provides the protection. CSF cushions the brain from contact with the skull when sudden movements of the head occur. It facilitates chemical stability. The CSF rinses metabolic wastes from the brain and the spinal cord and helps regulate the chemical environment. One way it does this is by removing excess hydrogen ions. It provides nutrients. The CSF provides the CNS tissues with some nutrients like glucose. This is all about the protection of CNS. Now we will move to blood-brain barrier. The capillaries in the brain develop differently from capillaries elsewhere in the body. The endothelium of the capillary walls has a thick basement membrane and is lined with the astrocytes to form a unique structure, the blood-brain barrier. The brain requires a constant internal environment to function normally. To maintain this, the blood-brain barrier acts to selectively allow certain molecules to pass and to keep others from reaching the brain. Before blood-borne substances can move from brain capillaries to reach neurons, three layers of the blood-brain barrier await them. The capillary wall endothelium, the thick basal lamina surrounding every capillary's external side, bulk-like feet of the astrocytes that are bound to the capillaries. Astrocytes cover the non-myelin portions of neurons and blood vessels in the CNS, forming a blood-brain barrier. This barrier allows astrocytes to regulate what can leave the bloodstream to enter the CNS, protecting the CNS from potentially toxic chemicals. Astrocytes also regulate the contents of the cerebrospinal fluid by absorbing excess neurotransmitters and potassium ions. The junctions between the cells of the blood-brain barrier are tightly fused and leak-proof, which separates the blood from the CSF. Small molecules in solution such as oxygen, glucose and alcohol can pass rapidly through the blood-brain barrier, but larger particles cannot because their movement is highly regulated. This protects brain tissue from toxic substances and pathogens to enter, but may also be a barrier to some potentially useful medications. Now let us see the ventricles of the brain. Interconnected cavities known as ventricles exist within the cerebral hemispheres and brainstem. They are continuous with the spinal cord, central canal, and also contain CSF. Within each cerebral hemisphere are large C-shaped chambers known as the paired lateral ventricles. They demonstrate the pattern of a cerebral growth and lie close together anteriorly. A thin median membrane called the septum pellucidum separates them. Each lateral ventricle is connected to the thin third ventricle, which is surrounded by the diencephalon via a channel known as the interventricular foramen. The third ventricle connects to the fourth ventricle via the cerebral aqueduct, which runs through the midbrain.